to get things started, I would like to introduce our keynote today. So Dr. John Farrell is a professor of aquatic and fishery science at the State University of New York College of Environmental Science and Forestry, and serves as coordinator of the AFS major in the Department of Environmental Biology. He also serves as founding director of the Thousand Islands Biological Station, known as TIBS, at an aquatic research station on Governor's Island in St. Lawrence River, and he works to promote the mission of applied science, education, management to guide informed policy enhancement of fish and aquatic resources. John received his Bachelor of Science degree from Cornell University and his Master's and PhD from the Environmental School of Forestry, but also spent a year in Canada, yes, John, at York University, including a memorable summer with Anne Rocky et al. At the Dorset Lab in Central Ontario. He's about to enter and begin his 32nd field season at TIBS, which is wonderful. He conducts research on warm and cool water fish ecology, but also works on topics including environmental monitoring, wetland ecology, lower trophic levels, invasive species, disease ecology, and habitat management. John has mentored numerous students, including AFS Hutton scholars, undergraduates, masters, PhD students, and postdoctoral fellows. John is a past president of the New York American Fisheries Society chapter and has been associate editor of the Journal in Wetlands and completed special issues of the Journal of Great Lakes Research, Hydrobiologia, and the North American Journal of Fisheries Management, and co-edited an AFS book on muskellunge or muskie management. In 2017, he received the award for exemplary researcher for excellence in mid-career achievements. John strongly advocates for field stations and marine laboratories and is active on the advisory board for the Central Michigan University Biological Station and the Cornell University Biological Field Station, where he started his research. And I often read these and I say to myself, wow, I need to do more in life. And John, that is no exception. So if you would be so kind as to share your screen, and we can start with your talk.
There we go. Now you can see the beautiful picture of the island. So moving along, I apologize for that, this technology stuff. But so this is actually a photograph of Governor's Island, where the Thousand Islands Biological Station exists. It's offshore of Clayton, New York, and it's really right across from Gananoque. And we live in the archipelago of the Thousand Islands, and the ecology of place is my theme today. So there's a really cool book. This is a book by Ian Billick, and some of you and Mary Price. Ian is the director of the Rocky Mountain Biological Laboratory, and they co-edited this book, and there's numerous contributions about place-based ecology, and that's the form of ecology of place that many of us come from in terms of those who do ecological research on long-term ecological research sites or monitoring aquatic and fisheries in specific locales for long periods of time. And I think it's just marvelous the insights that you can gain. So I really don't find it, I do find working all around the world, like my colleague Steve Cook, who I know is associated with the conference, working in all these various places, but focusing on a single spot has its appeal in terms of really trying to develop a depth of understanding and really trying to understand how a place works. And another theme is if you work on a place like the St. Lawrence River, which is at the base of the Great Lakes, what goes around comes around. So there's themes here with invasive species and how they're impacting ecosystems. And there's really, all of us might agree that there's no place, anything like this place, anywhere near this place. So this must be the place. So I got this from a website. If you're interested in like the broader and deeper understanding of the ecology of place, check out this website. It actually comes from a fellow from Southern Ontario, and it really gets into the depth of this concept, which is really gaining steam in urban ecology, but we're using it in the sense of ecological research and monitoring. So like I mentioned, I work in the Great Lakes system here in the upper river. It's an incredibly large basin, and I'm sure many of you are familiar with this. If you look at the basin in its entirety, it's like 1.34 million square kilometers. So it's the largest east-west drainage in North America, and the entire Great Lakes system, its natural outlet is through the St. Lawrence River on its journey to the ocean. So there are some diversions in the basin, but the bulk of the water is going through this system. So it's the largest freshwater body in the world, and it connects to the Atlantic Ocean. So it's just a fabulous place to do research and understand, like, connectivity is a really strong theme between the Great Lakes and the Atlantic Ocean. It's also a binational waterway. So this is, I was hanging out of an airplane with one of my colleagues who runs a plane called Fish Air, and will help us do aerial imagery. And it's just daunting to look at the archipelago of the Thousand Islands. There's like over 1,800 islands, and you can just see the complexity. So I took this photo quite some time ago, and it was just daunting. And I felt sort of first so impressed with the immense diversity of the system, but also just like I felt like we would never be able to understand anything about it due to its immense complexity. So the field station is a tiny island that's over here off of Clayton, and it's in this massive archipelago in one of the largest rivers in the world. So what a fabulous place to be. It also has a great diversity of fish. So I think roughly about 70% of the entire Canadian fish fauna can be found in the St. Lawrence River ecosystem. So it's just a wonderfully diverse set of fishes. And we really focus, because this is where the money is, we focus on the apex predators that serve as important sport fishes. So you'll see here the muskellunge and the northern pike, the assassids. We work a lot with centrarchids, the bass species, the walleye, yellow perch, and a whole variety of others. So we try to take a perspective. It used to be frowned upon when I began my career. People were trying to move away from single species management, but I make an argument that there's still a place for single species management as long as you're doing it in a holistic context, because you can maintain focus on trying to best manage a specific species. So the St. Lawrence River is also interesting because of its history of use. It's a what we call a working river, and the river is home to the St. Lawrence Seaway. So there's been numerous dams and locks created to harness this river for power, and also so it can serve as a conduit for trade with its international shipping. And the river over here on the left kind of shows this thread-like structure with some rapids. And this is a picture before the Robert Moses Saunders Power Dam came in. So as a Canadian US government initiative to create this massive public works project, which provides like 1,800 megawatts of power, which in the day was a really significant resource. It still is, but it created this massive area called Lake St. Lawrence. It's basically a fluvial lake, and that was due to the damming of the Long Sault Rapids here. So the river was much narrower, but this is downstream of the archipelago and the Thousand Islands. So we're significantly above this this change, but we interface with it. So in the Thousand Islands, the flow regime has changed somewhat, but the islands are actually, and the habitats are actually the same as they've been since post-glacial time. At the Thousand Islands Station, we kind of focus on these areas. This kind of represents our aquatic research program. So we're born out of working on this species, the apex predator, the muskellunge, and we've diversified over time since working in conservation of this species since the early 1980s. So now we do work in population and food web ecology, invasive species ecology, and also restoration ecology, working with habitat. And we do this in the context of a changing climate, a regulated flow regime, which you'll hear about, and the impacts of humans, which since COVID outbreak has happened, is really prominent. It's great to see humans using the ecosystems, but we are seeing more impacts on and uses of fisheries than ever before. Licensed sales in New York State for angling are up 30% this past year. So there's going to be additional strains on our resources, which we have to be cognizant of. And we also do a lot of fish counting. So I have a Dr. Seuss book here. I don't think this one was removed from the market, but 1, 2 fish, 2 fish, redfish, bluefish. We do a lot of fish counting, but we also do a lot of other research associated with fish in our monitoring program. This is one example. This is seining is kind of a tradition at Tibbs, and we began seining surveys back in the early 1980s, and then they became standardized over time. So this is one example of one of our standardized sampling approaches, 4-person to a fine mesh seine. We also, this is done in July every summer. Later in the season, we switch to a larger mesh seine that's twice the size of this one. And we've really, in the past, targeted the assastids, and we've picked a number of sites that we have standardized surveys in, where we do a total of 90 hauls, and then we have a series of exploratory sites. So 90 hauls represents that one survey. It's matched with an aquatic habitat assessment, so we assess the plant community using point quadrat methods. We do substrate analyses, monitor temperature, and we really look at the entire fish community. So this is a sample of about 150,000 fish in our survey of 56 species. So it's quite a diverse environment. We focus on the nearshore and geomorphic types in protected and open embayments primarily with this survey. And you can see there's even though we target assassids, they are really a tiny amount of the diversity of what we catch in this survey. 35% minnows, banded killifish make up a huge, like a quarter of the entire fish fauna, and then persids, yellow perch. When you combine all the centrarchids, it's about 18% of the community. And then there's a whole lot of other species in this small other category, which is this brown slice of the pie here. And I wanted to point out here, this is a new component of the fish community, which is a growing 5%. So when you think of when gobies became introduced into the system around 2005, 2006, now they're playing a bigger role. So this segment of the pie has been increasing, and it's had quite an ecosystem impact I'll discuss in a moment. When we look at some of these species, so we can track their dynamics in terms of the catch rates in our gear. We can study and develop hypotheses. So this is where the monitoring program really interfaces with research. For all of those who do monitoring, I think they know that it develops hypotheses that we can make about the system. It allows us to be proactive about management. So in this example, we can see that like blunt-nosed minnow, which is one of the most abundant species in the entire system, it has these significant spikes, and we've seen kind of a downturn in many of these species. Banded killifish, you can see quite a bit of variation in the catch rates, yellow perch as well. And we know from earlier studies looking at things like size spectra and fish community change, that the eutrophication of the Great Lakes had an influence on our fish communities, that now with drysinids and changes in nutrient dynamics, that we're seeing declines overall in fish populations throughout the region, with the exception of things like the round goby here in the lower left, where we've seen increases. You also see this like sawtooth variability with certain species, and there's something I want to show you here. You're probably aware that in the system, we just had 2 major floods. 2017 and 2019 were 2 major flood years, which had really strong influences on our monitoring program. So we know that seines are a depth-dependent gear, and I think here in 2017 we had to hire the tallest members of our crew to go out and tippy-toe and sometimes swim in order to manage to collect our samples. Well, you can imagine gear efficiency could have a real influence on the decline in these catch rates. But what's interesting is in some cases we see with the higher water level, we got actually higher catch rates for certain species. So we know that it's not just entirely a gear influence, but there's also behavioral influences that influence our catch rates. And it's something that with your monitoring programs, you really have to be cognizant of the factors that are influencing your ability to standardize and catch specific and be species specific about it. So that's the message here. So moving into a theme about talking about invasive species. So the St. Lawrence Seaway is really the poster child for aquatic AIS invasive species. We've had over 180 invasive species into the Great Lakes. I think about 60% of those are associated with ballast water and ships bringing in species from primarily Eastern Europe. There's a great volume of research on this, but on our end, when we see an invasive species come in, it's becomes part of a long-term assessment program to kind of understand the role and impacts of those species over time. And you probably recognize some of these customers. This is a Tubenose Goby, very recent invader. This is the round goby up here. We have Bithotrephes here, two types of Dreissenids, bloody red shrimp. This is the Cercopagus and Echinogamrus over here, an invertebrate. And we know this is in the lower river and we're on the lookout. So there's a tench working group looking out for European tench. So constantly on the lookout for invasives. This is a European frogbit, which came from Ottawa River in the 1930s, and it spread coast to coast. And we also see invasive typha. So this is a hybrid cattail. So some of these species have become really important in our research. Here's some examples of why. We have a native benthic fish here, which is the tessellated darter, Ethiostoma olmstedii, and this is the round goby. Sometimes I ask people to try to tell the difference, and it really is obvious for a fisheries specialist, but for the common person, it looks very similar. But what we see from our seining catch, that data I was showing before, is darters used to be really well represented in our seining data. They represented about 14% of the young deer muskie diet. And then since round gobies came in, we've seen a complete crash in tessellated darter. They're actually absent from all our surveys for 3 years. Last year, we actually caught one. So they're not fully extirpated, but they're very close to that. And the other interesting thing here is you see multiple surveys here. We got 2 seining surveys and one of our trap net surveys, and you can see they all kind of show a similar pattern, okay, with this really increase in rise and round goby, and then they all come crashing down during the floods again here in 2017. So we know that those floods had an impact on our catch rates for round goby. Round goby have changed the entire ecosystem. That's not news to anyone here. They eat invasive mussels. They have these trade-offs. So they eat invasive mussels. They're bringing benthic energy up into the food web. They increase fish growth. They buffer other native species from double-crested cormorant predation. So they're helping to balance the food web. Recent studies are actually showing that they create what's called a growth dilution, and they're reduced in contaminant levels. We've been studying that with a colleague with mercury accumulation, and we see this growth dilution. But on the other side, they're an egg predator. We know they displace native fish. They alter host parasite relationships through the enemy release hypothesis. They affect angling. And they're spreading to other systems. But a really big one that a lot of people don't think about is they serve as a vector for VHSV. So we have a PhD student who's focused entirely about how round gobies maintain VHSV in the system. We know there's about 100 variants now. You've heard of COVID variants. Well, there's about 100 VHSV variants in the system. And we're trying to understand how those are going to affect the future of potential disease outbreaks. So here's our fat bass. They're getting awfully close to the record. This was on the New York side just a few years ago. We saw bass prior to all these invasives, got a little bit bigger with dreissenids, and now they're just breaking the bank. So that's seen as a big positive. We also know that there's egg predation. So there's been quite a bit of research on egg predation. We recently added in other centrarchids to look at how nesting habitat selection influences their exposure to different predator communities. So there's other predators such as minnows that have big impacts on species that choose that as a spawning habitat and are nesting. The other thing to think about is broadcast spawning. So if you're a broadcast spawner and you have no parental care, you don't have that protection to guard the nest from a goby. And we recently ran some experiments that showed that yes, goby can find eggs in complex habitats, and the habitats did provide a degree of protection from egg predation, but even in the most complex habitats, 50% of the eggs in those treatments were lost in 24 hours to gobies. So broadcast spawning hasn't been adequately assessed in the field in these systems. We got a new goby I mentioned. This is another Canadian partnership. We worked with Matt Windle at the St. Lawrence Institute and combined our data, and we were looking at changes in the distribution of tubenose. Interestingly, tubenose and round goby were brought into the Great Lakes about the same time. You can see these cool little tubes on them, but it took the tubenose a lot longer to spread into the system. There was a single account here in 2011 here in Kingston, but in recent years, 2017, 18, and now 19 and 20, we see this eastward movement, and we think that it might be due to like secondary introductions and downstream drift. So we're seeing spreading now of tubenose in the system, and we're trying to study their impact. So there's a lot of like diet and isotope type studies going on. And you can see these tubenose, we only see like age 1s, and we see very few age 2 tubenose. So they're kind of a smaller animal, and they're more comparable to the small round goby. And the other point is that we're seeing in the upper St. Lawrence some of the largest gobies, round gobies that exist in the entire system, and maybe globally. So there's a few places that have these giant round gobies, and the St. Lawrence River is one of them. So we've been comparing like diet, stable isotope, and we're seeing that these larger round gobies get a greater proportion of their energy from dreissenids relative to the smaller round gobies and the tubenose, which are focusing more on plankton and invertebrates. So they have a greater diversity. But even all the gobies in the Thousand Islands region have a greater diversity of diet than what we see in other places in the Great Lakes. So this is work I'm doing with Roxanne Razavi, and we co-advised a student named Aman Paksad in our lab, and we're trying to understand about how these invasive gobies are going to be affecting our food webs. I'm going to take a quick shift here to working on our sentinel species, the muskellunge, Great Lakes muskellunge. It's just an iconic fish in the river. Like I mentioned, it's where we got our start with studying assacids and muskellunge in the system. It's really amazing because it's a functional management process between the U.S. and Canada. So we have a management plan. We actually meet. We have a working group organized under the Great Lakes Fisheries Commission. And when we started, this fishery was collapsing in the late 1970s, and there was very little information base to support management of the species. And so much has happened since that time through this binational effort. The plan, and it hasn't changed, we want to maintain muskie as a self-sustaining component of the fish community to protect its ecological role as an apex predator, and lastly, to have a quality trophy fishery. Okay, so numerous efforts like the Muskie Release Award through Save the River, critical habitat identification, increasing size limits, and now a voluntary catch and release have really changed the future for this species throughout its distribution in North America. So there's been a lot of progress made studying its early life stages through juvenile stages. And you can see this is a juvenile. They have this really cool cryptic coloration so they can hide in vegetation. So this is a fish species early in life that is really entwined with aquatic vegetation as an important habitat. More recently, I'll mention in a moment, we've been learning about subadults, and we're also monitoring many of these life stages to understand population processes. So critical habitats have been identified throughout the international St. Lawrence River on the both US and Canada side. Whether or not they're being protected is another question I can't get into today, but there is some level of protection. And there's been some interesting movement studies beginning with early radiotelemetry studies, the field studies I mentioned, but more recently with my colleague Steve Cook through Sarah Walton and Steve, a group of members in his lab and my lab got together, and we did a big hydroacoustic array study to look at kind of this missing life stage where it's subadults, the fish that are emigrating from these critical nearshore habitats. It was unknown where they went, and it was an important conservation issue that Sarah Walton did her master's project on and did a wonderful job. So check out that research. And it was just a very fruitful partnership. So all those management activities, sometimes we do regulations and it's kind of feel good. We just do those things. But in this case, we actually monitor and assess whether things are working using the data. So this is our TrapNet data set. And before and after those big regulation change and voluntary catch and release, we saw that classic fishing down of the population. So you could see that the population size distribution was small and for both males and females. And after those changes and how muskies were treated, there was a release from that pressure and exploitation. And we saw a greater increase in larger fish in the population, which was just a wonderful sign. I dug this out of my email. It's a long, long story, but John Castleman was a close colleague in this, who worked at Glenora and who is now retired but still active. And he said that all analyses of the large quantity of age and growth data that now exists for muskie of the upper river suggest that populations are strong and productive. So I pulled that out of my email, and that was in June 2003. And this is what happened in 2005. All of a sudden, we started, for no reason people could understand, carcasses floating all over the St. Lawrence River of dead muskie, really big adults. It happened for a 3-year period. And we saw this in our trap netting data. So after what I'm now going to tell you is related to VHSV, which was determined actually in a laboratory in Guelph from a fish from 2003, and then later by Michigan State and Cornell. And now they have quantitative PCR tools to test this in fish. But we saw basically losses of complete spawning populations in the river. It was really discerning and a sad story, especially on top of that deep conservation effort. And this is, the seining again is important, and it's designed to monitor young of the year muskellunge. So like I mentioned, each one of these data points in 2 surveys, they represent 90 seine hauls. Many of you in the audience might have helped us do this. You're welcome to come volunteer and check it out. But we do those surveys, and we saw, like, when the management was clicking, really high levels of muskellunge reproduction, and they have since crashed. It's getting quite, this past year was the first year in 180 seine hauls in quality muskie habitat. We caught 0 muskellunge in the survey. And we're very concerned about the future of the muskie population. We're also seeing it in the fishery. So this is the effort line here. In 2020, the effort went from an average just over 600 hours in our survey to over 2,160 hours of fishing effort in our database this past fall. So I couldn't even put that data point on the graph. And what's just concerning is those people only caught 16 muskellunge in our survey. So we had the highest effort we've ever seen, and we had the lowest catch rate we've ever seen in the same year. So people are very concerned about this iconic species in the river, and because we're seeing problems in the adult population. We had a disease that left a legacy. We're seeing collapse in the production of young, and now it's showing up in the fishery. So there's a big effort to try to restore the muskie population that's run out of tibs with lots of partners. We're raising fish, we're marking juveniles, we're replanting fish into habitat, and we're studying their population processes. Fortunately, there's genetic studies that have allowed us to do this kind of work. So doing restoration again. I know I'm running low on time, but I just wanted to mention another factor, and that is water levels policy. So, you know, we know in the St. Lawrence River that the water levels are managed, and that has a strong impact on the zonation of wetlands in the system, and how nutrient fluxes in the system occur, and connectivity across habitats. There was a water level management plan that is trying to release the level of constriction on the hydro period. So you can see how the management of the dam has constricted water levels. If it was unregulated, they'd be much more variable. So this has been an important thing that's been studying, and it's linked to the expansion of this non-native plant that's taken over much of the habitat in the system. So this is a really important area for research, because these are the critical spawning habitats for saucids throughout the system. We're seeing this trend of deep water spawning that's associated with that water level regulation and habitat change. There's efforts to try to change that. So I liken this to cardiovascular disease, where we're actually reopening arteries and connections so the fish species can reach the upper spawning habitats that they prefer for reproduction. And there's numerous projects going on throughout the ecosystem trying to reestablish connectivity and break up some of this invasive typa so it's beneficial to a fish. And we've been evaluating these projects to try to understand what's happening, and I work closely with my postdoc, JP LeBlanc, and we're seeing positive responses. In our surveys, we've caught over 400,000 fish coming out of these projects, and we can see really strong hydrologic signals that are driving not just northern pike, but also numerous other species. There was something like 44 species that are using constructed habitats, and we compare reference to constructed habitats to see if we can restore ecological function in our wetlands. There's been massive floods throughout the system that I mentioned, which really have brought into question whether Plan 2014 will survive. We'll have to wait and see on that. But I just wanted to finish up with a couple summary thoughts that our long-term data coupled with targeted research really allows us to better understand and hopefully help manage systems. Invasive organisms are really altering the community. They create a lot of uncertainty, as does the climate and water level change. But we still have native species that hold the top roles in the river. And we know that protection and restoration and management of habitat is our best hope for the future. So I just want to thank, our numerous sponsors and partners that work with us. Wouldn't be possible with all this massive effort. And I just thank you for your time today, and hope you have a great meeting.